Processes in recombinant DNA technology part 2. This is continuation of steps involved in construction of recombinant DNA and production of desirable product. This is, I repeat, is continuation. I was discussing isolation of desirable DNA fragment, how it is done. It is done by gel electrophoresis, and as you can read, the gel used is agarose. It is polymer of agar and porous in nature. It is used due to its porosity. Now coming to the next part, what is the what method is used? DNA is negatively charged due to presence of phosphate bond. So when electricity is passed in agarose gel, DNA tends to move toward the cathode according to the size of the fragment. This is used to isolate the desirable DNA fragment. Now what else we have to pay attention is DNA cannot be seen with naked eyes and in presence of normal light. That is why before starting this process, DNA is stained with, or we can say ethidium bromide is added with DNA. Now you can ask why. So first stepwise, if I discuss steps in gel electrophoresis. Number 1, DNA is stained with ethidium bromide. What it is? It is EDBr. Ethidium bromide. Next step, first let me tell the steps, then I will discuss further. DNA is stained, then DNA is loaded in the wells of agarose gel. Third step, electricity is passed, or electricity is supplied, or electricity is run. DNA fragments get separated according to their size, and when stained with ethidium bromide, that cannot be observed under a normal light. UV light or UV illumination, it's better to write DNA fragments are observed under UV illumination. Now you can ask why ethidium bromide is added in gel electrophoresis. Because DNA cannot be observed or seen with naked eyes as well as in visible light. So why EDBr is added with DNA during gel electrophoresis? Answer is to make it visible, as DNA cannot be seen in normal visible light and also with naked eyes. So along with EDBr, UV rays also used so that it can be seen. Here you can see the image showing. So these are the wells, largest, DNA band smallest, and one more thing is we must remember, just a moment, here I will write. largest, it's better to write smallest DNA fragments travel greater distance. So the largest are adjacent to the wells. So this is how DNA fragments are isolated and desirable DNA fragment is accumulated. Now coming to the third step, sorry, not the third one, fourth one, that is insertion of ligation of DNA fragment into the vector. That's the fourth step. Then preparation of competent step, competent host. Okay, so the fourth step, ligation of DNA fragment into the host. Okay, just a moment, I'll write. Fourth one, ligation of DNA into the vector molecule. Vector. So what is the vector? Vector is the carrier which carries the desirable DNA fragment. So isolated desirable DNA fragment is ligated with the vector, that is the carrier. Ligated with the vector using enzyme ligase. So ligase forms hydrogen bond in between the sticky ends which is produced by the restriction endonuclease, as I discussed earlier. It is produced by restriction endonuclease. Now coming to the next step, that is preparation of the host. Preparation of competent host. Okay, so how this is done, preparation of competent host? Just a moment. So just a moment. In case of normal cells, in case of normal bacterial cells, they do not prefer preparation of competent cells. Normal bacterial cells do not take up the isolated DNA fragment. So before ligation, as I told, ligation of DNA into the vector, it's easy. Then this vector must be inserted into the host cell, so that along with the vector, the desirable DNA fragment can multiply or replicate. So to insert the vector inside the host cell, first the host cells must be made competent. How it is made? So that they can easily take up DNA. Following are the steps. First step is number 1. Cells are treated with specific concentration of divalent cations like calcium. Cations, e.g. Ca2+, that is plus plus. Ca++. That's the first step. Second step, incubated on ice, then cells are incubated. Third step, briefly expose at 42 degrees temperature, 42 degrees centigrade temperature. This is called heat shock. Cells are heat shocked. Then fourth step, again put back on ice. These are the steps to make a cell competent so that it can take up vector DNA or any kind of DNA. Except this, also biolistics, gene gun, and one more method is given, that is micro injection is used. Biolistics or gene gun, just a moment, I'll write it. Biolistics or gene gun, micro injections are method used to insert DNA. Now how this is used? Micro injection is recombinant DNA is directly injected in the nucleus of animal cell, and biolystic or gene gun is used in case of plant. What it is done, that cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA and DNA. This method is called biolistics or gene gun. I repeat, cells are bombarded with high velocity microparticles of gold or tungsten, and these are coated with DNA. So microinjection is used in case of animal cells, and biolistics or gene gun is used in case of plant cells.